Howdy, folks. Thanks for checking in to MrUlrich'sLandofBiology.com. I am Mr. Ulrich. In this Notescast, we're going to be taking a look at my favoriteest of foods, cheese. Well, okay, actually, we're not going to be talking too much about cheese, but what we're going to talk about are the compounds that cheese is a good resource of, and those are lipids and proteins. A good place to start, if we're going to be talking about biochemistry, of course, is with carbon, that basic element of all organic compounds. Carbon and hydrogen, of course. Now, what's so great about carbon? Carbon is very, very good at making covalent bonds. In fact, it makes 4 single covalent bonds. Remember, covalent bonds involve sharing of a pair of electrons between 2 atoms. As you start to look at some of these structural formulas of various organic compounds, you'll notice that there are certain arrangements of particular atoms that are common from one molecule to another. We call these functional groups. They act almost like little sub-molecules. They have their own characteristics, which then they impart on the molecule which they are part of. A good example here is the carboxyl group. First off, don't get thrown off by the little letter R here. That's not a symbol for an element. There is no element R. It's a variable like X in math. It could be any variety of things, another organic compound, or just hydrogen. Then it'd be called methanoic acid because the carboxyl group can dissociate here and break that hydrogen off and act as a proton donor and is therefore acidic. So compounds that contain a carboxyl group are called organic acids. Likewise, the amino group here, we're going to see this in amino acids, which we're going to talk about later. It's where they get their name from, amino, and then carboxyl group is acid, but we'll talk about that a little later. The carbonyl group, we really talk more about when we get into AP Bio, so come see me in a couple years, we'll discuss that. This squiggly line is just like an R, same thing. It just means like it's attached to something. If it was an H, it would be methane. If it was an OH, it would be methanol. Speaking of OH, over here is the hydroxyl group. Well, this is the group here, of course, but what it's attached to is the R here. So if the R was a methyl, you'd have methanol. The phosphate group should be familiar to those of you who have taken reproduction and development already, as that is part of a nucleotide. Later on, next unit, we're going to talk a little bit more about ATP, and the phosphate is the P in ATP. Many of these organic compounds that we need to be familiar with are polymeric in nature. That means that they are chains of repeating units. And what we need to know about is how do we add more links to the chain? So how do we make polymers longer? Last lab, we cut out models of glucose out of paper and put them together. Built larger molecules by cutting an H off of one and the OH off of the other, making a water molecule, and then using those to open bonds, basically, to put them together like puzzle pieces. We taped them down onto a piece of paper. Because we are removing water to build large molecules, we call this process dehydration synthesis. Just like polysaccharides, proteins are also polymeric. They are chains of repeating units. Now, they're not saccharide or simple sugar rings in a row. These are amino acids in a row. But we're going to link those amino acids together by the exact same process. Hit that pointer again. There it is. All right. So in this case, what we're going to do is we're going to take 2 hydrogens off of this amine group over here. Lop an oxygen off of the carboxyl group, bond this carbon right to that nitrogen, and make what's called a peptide bond right here. So really what we end up with is called a polypeptide. You can think of it as instead of a chain of amino acids, which it is, it's also a chain of peptide bonds all in a row. But it is, again, through dehydration synthesis. If we're going to be familiar with proteins as chains of amino acids, then we should be somewhat familiar with amino acids. Kind of makes sense. An amino acid is a compound that has an amine group. Remember, we met that guy before, and a carboxyl group also before we met them. That's why this is called a amino acid, right? Because that carboxyl group is acidic. Now, each one of these amino acids, of course, is going to have the amine and carboxyl, amine and carboxyl. What gives each amino acid its kind of individual characteristics, je ne sais quoi, is what we call the side chain. In the case of alanine, that side chain is a methyl group, which we made a model of in a lab. Serine is a methanol group, and they have different characteristics. The characteristics are listed here in this table. These hydrophobic side chains are ones that don't mix well with water, like oil. Oil and water don't mix well. All of these are non-polar. And so amino acid, or excuse me, polypeptides, proteins that have these hydrophobic amino acids in them, side chains in them, that part of the polypeptide is going to try to rotate into the inside to get away from the water, since proteins are in cells and stuff like that, and cells are, watery. These other ones, these guys here, are actually really, they mix very well with water. And so those are going to rotate out of to the outside of the protein. And that kind of rotating is going to warp that polypeptide and bend it into a particular shape. The shape of the protein is so important. It makes up part of what we call the central dogma of modern biology. I know it says modern genetics up there, but this is a slide that I actually pulled off of a PowerPoint for the genetics. We'll see it later on. It's that important. Anyway, the central dogma goes something like this, these 4 boxes here. So we know that DNA is a polymer as well. It's a polymer of nucleotides, and each nucleotide has a nitrogenous base, one of 4 nitrogenous bases. So really what we end up with is a sequence of nitrogenous bases, those adenines, thymines, guanines, and cytosines, or A's, T's, G's, and C's. And that sequence of bases is directly translated into a sequence of amino acids. And just like we can think of DNA as being a sequence of bases, we can think of amino acids as a sequence of those side chains. And like I was saying before, the side chains are going to interact with their environment and interact with each other, causing the polypeptide to kind of warp in a particular shape as those side chains do their thing. That warping of those different amino acids is going to cause a particular shape in that protein to form, characteristic of that particular chain of amino acids. It's the shape of the protein that's going to allow the protein to do what it needs to do. If it's a structural protein, or if it's a transport protein, or if it's going to interact with some other molecule and cause chemical reaction to take place. So all the functions of the protein are really dependent on the shape of the protein. And actually, if we think about it, if we go ahead and change any of these things back here, we run the risk of changing the function of the protein. Now, a lot of people will describe the function of DNA as it makes me who I am. And that's not wrong. It's a bit of an overgeneralization, I think. What DNA really does, it's the instructions for protein construction, and your traits, who you are, so to speak, is really the sum total of all of the proteins, the cumulative effect of the proteins that you have or the proteins that you don't have, and how much of them you have, and when you have them and when you don't have them. And so it's really all of these different various functions of the proteins that are going to cause different traits. Like I have blue eyes because the protein that most humans have in their irises that causes their cells to make melanin, a brown pigment, is broken in my genes, in my family, in my ancestry, and so I don't make that protein in my eyes, and hence my eyes don't make melanin, and they are blue. But this is how we actually get from DNA to a trait. What do I really mean by shape of a protein? Well, here's a couple different diagrams of some proteins. And here, this is the polypeptide chain right here. So you can see how this one long continuous loop, right, folds into these flats and curls around, gets pretty elaborate. There's a lot to these protein shapes. But the shape of the protein, just like a key, the shape of the key allows the key to interact with certain locks and change them from lock to unlocked or vice versa. The shape of the protein is going to allow it to do what it needs to do. Now that's putting things together. We're also going to need to take these things apart because if we're going to eat proteins for raw materials for food, we got to take them, cut them up into their amino acids and then rearrange the amino acids in the order that we want them in for our proteins. So how do we take these things apart? Well, why am I showing you a diagram of dehydration synthesis again if we're talking about taking things apart? Well, what we're going to do is we're really just going to run dehydration synthesis backwards. We're going to stuff H2O back into the molecule, thereby forcing it apart into two separate molecules. This process is called hydrolysis, the hydro for the water and lysis for breakdown. I remember this by remembering that lysol works by breaking down the cell membranes of bacteria and germs. Also, a way to remember this is that when you go to a restaurant, quite often you'll be asked if you like to have a drink of water with your dinner. And part of that is because you need water to digest the dinner they're about to serve you. And that brings us to lipids. Lipids. If all the candy in the world just kind of went away, I'd be a little bummed, but I wouldn't be that bummed. I mean, I like chocolate. Chocolate, okay. But really, I don't have a sweet tooth. What I have is a fat tooth. I used to love to eat donuts. Oh, delicious, delicious lipids. Anyway, sorry. What the lipids that we're going to be talking about here are all based on glycerol. A glycerol is called a triol because it has these three OH groups. And it's onto each one of these OH groups that we're going to attach a fatty acid. Stearic acid here is an example of a fatty acid. So we're going to take 3 stearic acids. We're going to put one here and one here and one here. And by doing that, we're going to form what's called triacylglycerol or a triglyceride. Because we're making we're doing dehydration synthesis in 3 places here, we're actually going to be making 3 water molecules. So let's balance that equation. But lipids. Formulas. Okay. Here are some organic chemist structural formulas here. It's kind of weird if you think about these. These are organic chemists, so you think it'd be all about carbon and hydrogen, and it is, but they don't draw the carbons and the hydrogens in. They know where they're going to be. Right at each point here, this would be a carbon, here would be a carbon, here would be a carbon. So each one of these spots here would be a carbon. And it's assumed that you're going to know that each one of these carbons is going to have 2 hydrogens attached to it. This is going to have 1 hydrogen. That's going to have 1 hydrogen because it's a double bond, right? So they don't draw them all in. So this is a fatty acid here. And even though this has this like zigzag, right? Generally speaking, it is in a straight chain. If we're going to draw our best fit curve through this, it would be straight. Here's a triglyceride with the 3 fatty acids. And here you can see with this double bond here that not each one of these fatty acids is the same. And that does take place. So that's your basic triglyceride. Now down here, the phospholipid, this is really important. Instead of having 3 fatty acids, 1, 2, 3, it has just 2 fatty acids. And the third one instead is one of those phosphate groups, hence phospholipid. These are very, very important. We're going to talk about these next unit when we talk about the structure of the cell membrane, as these make up the bulk of the cell membrane itself. Now up here I have cholesterol. It certainly doesn't look like any of these guys here because it's pretty different, but it's still a lipid. We don't spend much time talking about cholesterol as a lipid in Regents biology. Come see me again in a couple years in AP Bio and we'll talk more about cholesterol. All the triglycerides have glycerol. So that's going to be the same in each triglyceride. What makes one triglyceride different from the other is going to be the nature of the fatty acids that are attached to the glycerol itself. And these fatty acids can be classified in two categories. And therefore, the lipids that they're part of can be also classified in those same categories, saturated versus unsaturated. Now, what are they saturated with? They're saturated with hydrogens. So a saturated fatty acid in that hydrocarbon chain is going to have as many hydrogens as could possibly fit. So each one of those carbons is going to be totally filled up with hydrogen. That means that there aren't going to be any double bonds between the carbons. If we look down here, you can see let me get my pointer on, there we go. Right here is a double bond, and that leaves us some space here for more hydrogens. Up here up top, there are no spaces for the hydrogens. It's totally saturated with hydrogen. That gives this a straight chain, right? Even though we know it zigzags like it did with in the organic chemist structural formulas, the best fit curve again would be a straight chain. Because those legs are straight, that means I can pack those lipid molecules together a little tighter. I can fit more into a space, giving them higher density. Because of the packing, they also tend to be solid at room temperature. And so these saturated fats are referred to as fats. Most of these fats are going to come from animals and animal products, which makes them kind of expensive when we compare them to the unsaturated fats. And let me get there. All right, so if saturated fats are saturated with hydrogens, that means the unsaturated fats have room for more hydrogens. Again, here's that space where we can put those hydrogens. That space is allowed because it does have a double bond between the carbons. And that gives the chain here a little bend. Right, and if you look up here, here would be one double bond, here's another double bond, and arachidonic, there's a whole bunch of double bonds kind of in a row there, giving it a little hook shape. Because of these kind of weird angles, it's harder to pack those fatty acids together, and that means that these lipids, unsaturated lipids, have lower density and are liquid at room temperature. So these are the oils, and they come from plants. And because if you're going to grow animals, you're going to have plants to feed the animals. That makes animal products far more expensive. So if you're a candy bar maker and you really want to make a cheaper candy bar, you don't have to go to the saturated fats for your solid lipids. You can take the oil and heat it up and bubble some hydrogen gas through there, weaken those double bonds and cram some hydrogens back in there again, and you end up with a hydrogenated oil, which is solid at room temperature and cheaper. That's enough. We'll stop there. Thanks for checking in to MrOlicksNanobiology.com. If you have any questions, you can certainly leave them in the comments if you're on YouTube, or by all means, send me an email with questions from this notecast or any other notecast. Other than that, see you in class.